Some directions in vector space are special. Push a building along its resonance frequency and it amplifies. Push perpendicular and nothing happens. Eigenvectors are these resonance directions. Tie a matrix to most vectors and they rotate and scale, they change direction. But eigenvectors only scale. At times v equals lambda times v. The vector v doesn't change direction. Lambda, the eigenvalue, tells you how much it stretches. In PCA, the eigenvectors of the covariance matrix are the principal components. The eigenvalues are the variance explained along each component. PC has the largest eigenvalue, the direction of maximum variance. Any symmetric matrix can be rebuilt from its eigenvectors and eigenvalues. Stack the eigenvectors as columns, multiply by a diagonal matrix of eigenvalues, multiply by eigenvectors transposed. This is spectral decomposition. Drop the small eigenvalues, and you get an approximation. The top k components capture most of the structure. This is exactly what Pitha does. Keep the top 50 eigenvectors, discard the rest. It generalizes eigendecomposition to any matrix, not just square, not just symmetric. Any matrix equals U times sigma times bit transpose. U contains the left singular vectors, patterns across cells. V contains the right singular vectors, patterns across genes. Sigma is a diagonal matrix of singular values. How important each pattern is. Image compression. Keep the top 10 singular values and reconstruct. The image is recognizable. The data is 99% smaller. Gene expression. Keep the top 50 components. The biology is preserved. The noise is gone. The connections cascade. Here is eigendecomposition of the covariance matrix. UMAP starts from PCA output. Non-negative matrix factorization decomposes the expression matrix into non-negative parts, metagens. The Jacobian matrix of a node system as eigenvalues that determine stability. Negative eigenvalues mean the system returns to equilibrium. The graph Laplacian's eigenvectors define spectral clusters in networks. Linear algebra is everywhere. 20,000 genes, but only 50 principal components capture 95% of the variance. The data lives on a low-dimensional manifold embedded in high-dimensional space. Why? Because gene regulation is modular. Hundreds of genes respond to the same transcription factor. Thousands co-vary because they belong to the same pathway. The low-rank structure isn't a trick, it reflects biology. The architect's blueprint had 20,000 lines, but only 50 independent design decisions. Eigenvectors find the resonance directions. Eigenvalues measure their strength. SVD decomposes any matrix into patterns and importance. Low-rank approximation compresses without losing biology. This is the architect's toolkit. Every method in computational biology, from PZ to neural odes, is built on these foundations.